Namaskar. Welcome to the Bengali Association of Greater Chicago's Health and Wellness Seminar Program. I'm Deepak Ghosh, coordinator of the seminar programs. Tonight we have a very important and timely program and topic. The topic is latest on the COVID-19 pandemic issues, with particular emphasis will be given on the dangers of new Delta variants and other variants, booster shots, vaccine for younger children, and how long the current vaccines last, etc., and many more that will be covered. We have a great speaker for you tonight. Our speaker is Dr. Archona Chatterjee. She's an MD, PhD. She's the Dean of Chicago Medical School and Vice President of the Medical Affairs for Rosalind Franklin University of Medicine and Science. For the past 7 years before she joined the Chicago Medical School, she served as professor and chair of the Department of Pediatrics and senior associate dean for the faculty development at the University of South Dakota, Sanford School of Medicine. Dr. Chatterjee has served on several national advisory boards and committees, including FDA's Vaccine and Related Biological Product Advisory Committee and the Pediatric Infectious Disease Society Board of Directors. Trained as a pediatric infectious disease specialist, Dr. Chatterjee has practiced in her field for over 20 years, conducted over 120 clinical trials. She has published over 90 peer-reviewed scientific articles in national and international journals, given over 95 media interviews, and published over 30 newspaper articles. What a great accomplishment. Some of you might have seen Dr. Chatterjee in the local or national TV appearing as an expert speaker. Dr. Chatterjee has accomplished another great milestone. She is the first female medical school dean in the whole nation with reported Indian descent, according to the Association of American Medical Association, or AAMC. More importantly, or equally importantly, she is an active member of BAGC. We are extremely proud of Dr. Chatterjee's accomplishment, and we are really honored to have her as a speaker tonight. So now, ladies and gentlemen, it's my distinct honor and privilege to introduce to you our speaker for tonight, Dr. Archona Chatterjee.
Thank you very much, Dr. Ghosh. Namaskar, and hello to everyone who is here in the audience today. Before I begin, I do want to acknowledge my niece, Jayati Datta, who is actually joining from the UK. So I'm very happy that a family member of mine that is so far away is able to join us today. Without further ado, we will go ahead and get started. I will share my screen here in a moment, and you should be able to see my slides. I'd like to thank BAGC for this opportunity to be with you today and to share the knowledge and expertise that I have in this field that hopefully will be helpful to you in understanding what is happening with this worldwide pandemic and how we can control it. So without further ado, let's move ahead. The first thing I need to do is read the statement related to the FDA, because as Dr. Ghosh mentioned, I am a member of the FDA's Vaccines and Related Biological Products Advisory Committee. Any information I provide and my opinions on requested questions are completely based on my own personal and professional knowledge, understanding, and expertise only. My expressed opinions on the vaccine's approval process do not necessarily reflect the opinion of either the Vaccines and Related Biological Products Advisory Committee, also called VRBPAC, or the FDA, to which the committee provides only advisory services, and they should not be misconstrued or misrepresented. Also, this information does not directly or indirectly bind either the committee or the agency that is served by the committee. Lastly, I am required to complete the FDA's conflict of interest screening forms provided before each meeting of the VRBPAC. So with that out of the way, let's start by looking at what makes the virus that causes COVID-19 so pathogenic, so easy to spread and cause disease. First of all, I want to call your attention to the picture that has the red circle on it. That is showing you the binding site where the spike protein, I'm sure you have heard in media reports, read about the spike protein. This is the part of the virus that sort of pokes out on the outside. This spike protein is what attaches to the human receptor. It's like a lock and key mechanism. And so this is the area in which the mutations occur. And because of those mutations, the virus is able to escape treatments and vaccines, et cetera. So this is a very important interaction to know. And part of the reason that the virus can cause disease is because it binds very tightly to this human receptor. Another thing that this virus does is it actually blocks immune responses that exist naturally in our bodies to viral infections, and then alters other immune responses that occur as a result of an infection. So it's because of this aberrant immune activation that you see many of the symptoms. So the virus itself actually doesn't cause symptoms. What causes the symptoms is the response to the virus. So high fevers, body aches, headaches, the flu-like symptoms that you see are all actually because of the immune response that is occurring. And many of the more serious complications that you see, such as respiratory failure, also occur because of that immune response. Now, one of the things that has been discussed a lot is the development of the vaccines. And one thing I wanted to focus on is to help the audience understand that there is a process by which vaccines are developed, other vaccines are developed, which takes somewhere in the range of about 10 years, actually. So you start off on the left side of this graph, or of this picture, to show you that we first have to find a vaccine target. There is a preclinical stage of development, animal studies are done. They then go into clinical development, where there are 3 phases of clinical trials. You don't need to know all of the details of this, but the reason I'm presenting this is to help you understand that there is a whole long process lasting over years that takes place before vaccines are developed. Now, what we were faced with last year was a deadly pandemic for which there wasn't time to do many of these things in the way that they were done before. But what I want to assure this audience is that the steps were actually taken that led to safe and effective vaccines being authorized for use. So if you look at this slide, you will see that some of this exploratory work had actually been done ahead of time, because you will remember that there were other similar viruses, cousins of this virus, the SARS virus and something called the Middle Eastern respiratory virus, or MERS virus. And so some work had already been done, the animal studies had been done, et cetera. When it came to the clinical trials, instead of doing phase 1, then phase 2, then phase 3, they kind of telescoped these together so that you could have overlapping phases. And phase 1 and 2 trials were done together. Phase 2 and 3 trials were done together to shorten the amount of time that it took to collect all of the clinical data that we needed. And then you get into the regulatory review piece of it, which is where the agencies of the government, the FDA in the US, the EMA in Europe, et cetera, that actually approve and authorize vaccines come into play. Now, one of the things that the vaccine manufacturers did, and here's where the governments also come into play, is that they decided to do what is called production at risk. What this means is that they actually started making these vaccines before we had any data from the clinical trials. They said, we'll have it ready so that when we have the clinical trial data, if the data look good, we can release the vaccines right away. And so the governments bought the vaccines, the manufacturers made the vaccines, they had it ready to go. So that's why if you think back to last December, as soon as the FDA gave authorization to the very first vaccine, which was Pfizer's, Pfizer was able to start shipping that vaccine within a couple of days, and they couldn't have done that if they didn't have it ready. So this is what helped them compress that 10-year time period into a 10-month time period. Now, this is another long slide that talks a lot about this process and the US FDA. I'm not going to go through all of these, but just explain very briefly to you the difference between an emergency use authorization, which is called an EUA, and a biologics licensure application, which is called a BLA. So, you know, people use these terms intermittently, interchangeably. So they will say the virus, the vaccine is authorized or the vaccine is approved. Actually, technically, they mean two completely different things. So the vaccine is authorized, it's under an EUA. This is a special law that usually wasn't used for vaccines, but it was used by the FDA last year, given the deadly pandemic that was going on. So they wanted to get it out quickly. And the reason they were able to do that is because through this EUA process, the product may be effective, its benefits must be known to outweigh the risks. But other than that, you don't need all of the data that we need for a BLA, okay? So when people say that there were corners cut or this was done too quickly, the answer is there were no corners cut. It was done much more rapidly than it normally is. And what we did was we looked at this interim analysis of the clinical trial data. And based on that, we were able to provide recommendations to the FDA to provide authorization of the product, and it could be shipped out very easily. Another thing that's done is for a BLA, the actual manufacturing facilities have to be inspected, which is not required for an EUA. So hopefully you understand the difference between these two mechanisms and why the EUA was used as opposed to actual full approval and licensure. Now, most of you know this already, the major vaccine candidates that are authorized in the US, and I'm only going to speak about the US, even though I know that there are listeners from other countries that are here tonight. And that's because these are the data that I know the best. So the mRNA-based vaccines, there are two of them, Pfizer that I mentioned before and Moderna's. They're given in 2 doses. And then there is a viral vector vaccine, which is Johnson & Johnson or Janssen's vaccine, and that is a single dose vaccine. Now, again, we're going to get into some technical stuff here, so bear with me, but just to help you understand what actually happens, how are these vaccines actually made and how do they work? So the RNA vaccines, RNA is ribonucleic acid. It is a molecule that carries a message, okay? It is a code, if you will. That code is for that spike protein that I told you about earlier. And you cannot just inject RNA because it gets destroyed by your body very quickly. So in the vaccines, the RNA is actually enclosed in a little fat bubble. We call it a lipid nanoparticle. That is where the RNA is sitting. Once it is injected and it gets into your cells, the lipid nanoparticle opens up, the RNA is released, and the cellular mechanism, your own body's cells begin to make the protein. That's what's called the antigen. That antigen is then seen by your immune cells, which is what's shown at the bottom of the slides. These T cells, B cells that are part of your immune system actually begin to react, make antibodies, make immune memory cells and things like that. Now, you will notice that in this process, the RNA never goes into the actual nucleus of the cell. There's, excuse me, a lot of misinformation out there that the RNA can somehow get into your DNA. It actually cannot do that. You need a special enzyme. It's called a reverse transcriptase to do that. And our bodies don't have that enzyme. So the RNA actually never enters the nucleus. Therefore, it cannot become part of your DNA. So if you hear that from anybody, that is wrong. The next kind of vaccine that I want to talk about is the Jansen vaccine. This is a little bit different mechanism that is used. In this case, the carrier is not that little fat bubble. The carrier is another virus. And in this case, the virus is an adenovirus. This is what we call a non-replicating adenovirus, which means that this adenovirus, it gets into your human cell, as you see shown in the picture on the right, but it is only carrying that mRNA from the COVID virus, and it's actually not becoming part of anything within your body. It releases that mRNA, and then the rest of the process is exactly the same as I described before. Now to some common questions that we get with regard to the topic of COVID. So I'll go through each of these, and then we will take more questions from the audience as we go along. So the first thing is about side effects and reactions. I know many of you may have been vaccinated. Some of you have probably had reactions to the vaccines. I can tell you that in my family, my husband and I both got both doses of the Fisa vaccine. Neither one of us had really much of a reaction to either one. Our daughter, who is in her 20s, had the first dose and had no problems with it. And then with the second dose became really, really sick. She had fever, she was nauseous, she had lots of flu-like symptoms. So that is not uncommon, especially in younger people, to see those kinds of reactions. The important thing to know is that these are transient. It lasted a couple of days. I have a nephew who also had very similar reactions after the second dose. And again, he felt miserable for a couple of days, but got better and just took some over-the-counter fever-reducing medications and was better. The important thing to know is serious side effects, serious reactions, things like that. You'll see a whole list here of different agencies, different mechanisms by which these can be reported. The CDC, the Centers for Disease Control and Prevention in the US, actually put together a new mechanism, the vSafe mechanism. This is a cell phone-based app that people could report reactions through. And I never had to use it, but I have heard from people who have that it's very easy to use and they can just report their reactions based on that. The others are older systems that have been in place and are very effective for us to be able to study serious adverse effects and reactions. As far as how good these vaccines are, again, you are seeing a very complicated slide here. I'm not going to go through all of this, but just going to show you that as the clinical trials were completed, the data were reported in peer-reviewed journals. This is from the New England Journal of Medicine. And this is the very first vaccine, the Fisa vaccine, that the data were reported on in December. What you're seeing on the right-hand side of your slide is the difference in the cases that accumulate after the first dose of the vaccine were given. At the bottom of the slide, almost like a flat line in red, is the people that received the Fisa vaccine. And in blue, what you're seeing going up almost in a straight line is the cases that were accumulating in those who got a placebo, a saltwater injection instead of the actual vaccine. So it doesn't take a technical expert to see the big difference that there is between those who got vaccinated and those who didn't. This is another depiction of the same thing. Basically, the vaccine share of cases, if you look at the top of the slide, you'll see in the purple, the vaccine share of cases began to decline after about a week of the first dose. So even with the first dose, we began to see fewer cases in those who were vaccinated. And at the same time, you were seeing cases rising in those who were not vaccinated. And so the vaccine efficacy, of course, increased, particularly after the second dose. These are the data from Moderna. And again, this was published in the New England Journal for their vaccine. You're seeing a very similar graph to what you saw before in the middle of the slide. The vaccine recipients are at the bottom there. They did have a few cases, but those are very slow to accumulate, whereas those who got the placebo or the saltwater injection, those cases went up very, very quickly. This is another picture showing you pretty much the same data in a little bit more detail. These are the data from Jansen. And once again, you see the separation of the graphs. At the bottom in the darker blue are those who got the vaccine. In the grayish blue color at the top are those who got the placebo. And you see that people who got the placebo had far more cases occurring as time went on compared to those who had been vaccinated. Now, I get a lot of questions from people about comparisons between the vaccines. Which one's better? Okay, we actually can't answer that question because these vaccines were not studied at the same time in the same population. We call it a head-to-head clinical trial. Those have not been done. But I did want to show you a little bit about the differences. So between Pfizer and Moderna, these are both mRNA vaccines, right? But you can see on this chart that the dose is a little bit different. The dosing interval is different. Vaccine efficacy is pretty similar. The numbers are a little bit different in terms of storage, and the reactions are very similar, etc. So you can actually make some comparisons, but in terms of safety and efficacy, you really can't compare and say, well, this one's better or this one's worse. And then for Janssen's vaccine, these are also on this slide. You can see that the dose is 0.5 ml. The vaccine efficacy here is a little bit lower than what we saw with the mRNA vaccines, which are in the 94-95% range. But again, when people ask me, well, aren't those vaccines better? Mathematically, yes, but from a vaccine science standpoint, no, you really can't make that claim. And then we also want to see, okay, fine, you're showing me data from the clinical trials, right? How do these vaccines perform once we start giving them to millions of people? So this is a study that was published actually from Israel, where they had given almost 500,000 people the vaccine, and they looked at effectiveness of the vaccines in this much larger study. In the clinical trials, there were only about 30,000, 40,000 people. So this is a much, much bigger study. And what you find is that the vaccines performed extremely well. We have a number of new studies now that have come out since this one came out. This was the very first one that reported it, but the vaccines are very effective, and that not only reduced symptomatic disease, but hospitalizations and deaths, which was a very important thing for these vaccines to be able to do. So as I mentioned, we should not be comparing these vaccines because the trials haven't been done like that. And one of the things that people don't realize is as this virus is mutating, we start to see the efficacy of the vaccines changing. So for example, last week I saw a trial that showed that with the Delta variant that Dr. Ghosh was talking about, the Fisa vaccine efficacy, which in the original version of the vaccine which was studied, it was 95%, it was down to about 84%. So the efficacy can change depending on how the virus changes. Another question we get very often is, okay, how long is this immunity going to last? Do we need to get booster doses? The correct answer to this question really is we don't know yet. And that's because we have not had enough time to know whether the immunity will last for a year or for 2 years or 5 years. No one really knows that yet. There hasn't been enough time. What we do know is that the antibody levels do decline over time. So again, I saw a paper this week that showed that the antibody levels can go down by as much as 50%. However, we don't know what the protective level of antibody is. So even if the antibody has become half of what it was, maybe it's still effective. How do we know? If you're not becoming sick, it's probably working, okay? The other part of the immune system that we are not able to measure as easily is those memory cells I talked about earlier. And so there's cellular immunity as well as these antibodies. And we really can't answer this question about boosters yet. You might have heard in the media that there are stories about, you might need a booster dose if you're over 65 or if you have some sort of immune deficiency and things like that. So far, we have no indication that that is actually going to be required. And some of you may have seen the WHO request to actually not give people booster doses because there's a shortage of vaccines around the world, and that in fact, people should wait to get a booster dose until other people have a chance to get the vaccine. Now, what about these variants? Everybody's talking about variants, right? Will we need vaccines that will need to adapt to deal with the variants? The answer is probably yes. And in fact, many of the vaccine manufacturers have already announced that they are working on newer versions of the vaccines against the Delta variant and against emerging variants. The important thing to recognize is that with these new technologies, especially the mRNA and the vector-based technologies, you can actually make these new vaccines very, very quickly. It doesn't take as much time as it used to in the past when we had to use some older technologies. Now on this slide, you will see a little bit of a comparison between the different vaccines. And again, I'm not going to go into the details of all of this. I want to call your attention to the bottom right of the slide, where it's showing you the efficacy of the Johnson & Johnson vaccine, okay? And you will notice that it had an efficacy of about 72% against what was then called the US UK strain. This is actually the alpha strain that they were calling it. And then you'll see that the efficacy went down to about 57% against what was at that time called the South African strain. So this is actually the Gamma variant that is there. So you see some reduction in the efficacy of these vaccines, as I pointed out earlier. Now, what about special situations? People who have had COVID, okay, they had a positive test or they got treated for COVID. Should they get vaccinated? The answer to this question is a resounding yes. I have family members, my brother's family, a number of them were infected, my sister's family, I've had a niece who's infected. I recommended that they all get vaccinated. And the reason for this is because we didn't know how long the immunity from natural infection would last. We have a study that came out just yesterday, which showed that people who have had COVID, if they are unvaccinated, their risk of catching COVID a second time is actually 2 times higher compared to those who got vaccinated. So if you have someone you know of who has had COVID, please ask them to get vaccinated. The CDC does say it's a good idea to wait 90 days after the infection or treatment, so that we know that much time they're safe, but after that they should get vaccinated. We also get questions from people about, mixing up the doses. I hear from friends who have had family go to India. They got a couple of doses in India. What do they do when they come here? Should they get revaccinated? All of that stuff. Or they got 1 dose in India and now they're here. Which vaccine should they get? Because we don't have the vaccines that they're giving in India. Ideally, you want to give the vaccines as they were tested and studied. Okay, so if they're given in 2 doses, 3 weeks apart, then you should give it like that. However, we know that because of shortages, sometimes the time has been lengthened between the vaccines. This mixing and matching also is not a good idea, but if you have no choice, okay, you were in India, you got 1 dose there, you came over here, we don't have those vaccines, get whatever you can get and get the next dose if you need it. Dr. Ghosh talked about children. We now have at least 1 vaccine, the Pfizer vaccine, which is authorized for use in children 12 years and older. Unfortunately, only 1/3 or so of the US children that age have been vaccinated. So there's a big hole there. And for younger children, currently we don't have any authorized vaccines. However, those clinical trials are ongoing, and we're hoping that we will have those vaccines available within the next few months to vaccinate younger children. About pregnant women, we were in a discussion yesterday about this with some friends. It is very important that pregnant women get vaccinated. Unfortunately, there's a lot of misinformation. Sometimes their doctors are not telling them to get vaccinated. The CDC has made this recommendation several months ago because we know that pregnant women are at risk. Their infants, their babies are at risk of catching this infection. And it's a very good idea for them to be vaccinated. So they're not only protected themselves, but their babies are protected through antibodies that they get through the placenta. There are some people who have allergies, they ask questions. I have egg allergy, I have respiratory allergies. Those are really not much of a problem. But anyone who has a severe allergic reaction to any of the vaccines or any component of any of the vaccines should not receive any more doses. And it's a good idea to consult with an allergist if you're worried about allergies. And then there are people who have underlying medical conditions, diabetes, asthma, cancer. What do they do? Well, for most of these conditions, as you know, COVID is a very bad disease and actually can cause increased rates of hospitalizations, ICU admissions, and deaths. So it's actually very important for them to be vaccinated. However, if someone's had a transplant or they're taking chemotherapy or something, they should consult with their physician first to make sure that it's safe for them to be vaccinated. Another question I get very often for the clinical trials is, were there people of my community in these clinical trials? And I just wanted you to know that for the 3 vaccines that are in use in the US, Asians were included, and you see in red the highlighted proportion of Asians that were included in those vaccine trials. The important thing is that in the trials, there were no differences seen in the efficacy or safety side effects, no differences between people of different racial and ethnic groups. And it's important to have that information so people can feel safe when they're getting these vaccines. The last item I'd like to talk about is about building vaccine confidence. You hear a lot about people talking about vaccine hesitancy. We like to focus on the positive side is people who are concerned or hesitant, as some people say. How do we reach them? How do we build trust among them? Well, one of the things is to make sure that we empower people with knowledge so that they get the information correctly from within their community. And I think that's what BAGC is doing here, is trying to inform the community from someone who actually knows something about this. And, you know, there's a lot of social media stuff that's out there. A lot of it is bogus, you know, it's incorrect information, and people are just spreading that information. So what we want to do is spread accurate, up-to-date information. As you saw, I quoted you some studies from this week, one from just yesterday, because that is what you want to be doing. You want to stay on top of this and not be talking about things that might have happened months or even a year ago. It's important to make sure that people get vaccinated, that they understand the reasons for vaccination. And that's frankly the only way that we're going to get ourselves out of this pandemic. Without that, it's going to be very difficult to do that. Just wanted to say a word about the media, because it's not just social media, but even, you know, regular TV, radio, newspapers. So they get a lot of things right. You know, they've recognized that it's a dangerous virus, that the vaccines are safe and effective, and they've talked about mitigation measures, mask wearing, hand hygiene, social distancing, all the stuff that's needed, right? They get some things wrong too. And here are a few. I mentioned one of these before because they keep saying the COVID vaccines are approved, and they're not. They're all authorized at this point in time. None of them are approved, actually. They have also heard people say, well, this vaccine's better because it's 95% effective. This one's only 75%. That's not correct, as I pointed out. There are no head-to-head trials, no comparisons. You really can't say this. And unfortunately, this has been politicized a lot. So you hear a lot of people giving very slanted views, which are based on politics, not on medicine, science, and public health. And the last thing is that often you will hear them say, well, you know, my mother's cousin had this thing. Well, that's an anecdote. It's an interesting story, but those are not data. Data are based on science and on trials and studies, and they cannot be equated or compared to what individual experiences are. If you want to get more information, these are the websites, and I would request that the BAGC folks actually share my slides so that you can take a look at these websites. They will give you accurate information, up-to-date, correct information, and not misinformation and disinformation. I just want to acknowledge my family. They are here on this slide. My husband, Samar, who is listening in today, and my daughter, Maya, who is a medical student and is sleeping at the moment because she's very tired and she's getting ready for a set of big exams next week. With that, I am going to conclude my portion of the presentation. Again, a very big thank you to the audience for being here tonight. And I'm happy to answer any questions, or at least try to answer the questions you have. I'm going to hand it back to Dr. Ghosh. Thank you very much.
Thank you, Dr. Chatterjee. That was great presentation packed with a lot of good information. I'm positive our audience, BAGC members, will be benefited with the information you have provided. Now we'll go to the question answer, and there are a couple of ground rules. So we run through the question answer very smoothly. Number one, all your audio and video, particularly audio, would be on a mute position. If accidentally it gets turned on, please mute yourself. Otherwise, it creates a lot of disturbances, background noises. Second, if you have any question, then if you know how to go to the computer and raise your hand, please do so. We'll recognize, and our technical team who are watching, they will unmute you and you can ask the questions. And last but not the least, I would sincerely request, when you ask a question, please make it to the point, make it relevant, and not to make a big statement, whether it's a personal, political, or even scientific, because we have a great speaker. Let's hear from her. With that, I have already seen some of the questions I'm watching. So the first one, we're going to go to Fisa Chatterjee. Bhaskar, if you can unmute Fisa. Fisa, go ahead with your question.
Deepakta, I'm not able to see Fisa Chatterjee. He was there before, but it's not visible now.
Oh, uh, uh, I saw just a second ago. He sent me. Okay, we'll go to the next person, and if Bishu joins, let me know.
Sure.
Dr. Amit Mitra. Uh, Dr. Amit Mitra, go ahead. Unmute yourself, or Bhaskar, please unmute.
We have two raised hand, Deepak Chatterjee and Monotosh Banerjee. They will be in the next probably.
Okay, but
Whatever.
let's go to Amit. Amit, he already sent me indication that he has a question.
Yes, can you hear me?
Yeah.
Yes, Dr. Mitra.
And you can start your video as well if you choose to. Up to you.
Yeah, I'm not that familiar. Okay, I see that video. Okay.
By the way, while Amit is trying
Probably you
to,
can see me now. I think, yeah. Thank you, Dr. Chatterjee, for this timely presentation. I think I saw you at the advisory committee meeting, and it was excellent advisory committee meeting that I attended myself. But a couple of questions. Those are related to the vaccination in children, especially as you mentioned that recently FDA gave the emergency authorization for Pfizer vaccine in adolescents between the age of 12 to 15 years or 12 and older based on the risk benefit profiles. I was going through the warning section of the packaging insert and I saw that increased risk of myocarditis and pericarditis, particularly after the second dose. Would you please elaborate on these symptoms for these
Yes.
disease states so that the
Yes.
emergency treatment could be sought by the parents if needed?
Yes, so these are serious complications that have been noted to occur following, as you correctly pointed out, the second dose mostly. They are quite rare, they're infrequent, and most of the children, and it actually occurs in adults as well, it doesn't just occur in children. Most of the individuals who were impacted in that way actually recovered without serious complications following the occurrence. But when it happens, obviously, it's a very serious thing and people get worried about it. Usually the patient will experience fever, maybe some chest pain, difficulty breathing, etc. So those are serious symptoms. So anyone who is vaccinated who starts to have those kinds of symptoms should definitely contact their healthcare provider immediately and let them know that they have been recently vaccinated so that the physicians can take care of them appropriately.
Should they be rushed to the emergency room?
I think it is best that the situation be managed based on the symptoms at the time. So, you might say you want to rush to the emergency room, but if you've ever been to an emergency room, you know that you might land up there and then be sitting there for 4 or 5 hours before somebody takes a look at you. So it's a good idea to call your physician first so that they can assess how serious something is, and then they can actually call the emergency room to alert them so that when you get there or you take your child there, that you get attention immediately.
Thank you for this information. My second question has to do with the authorization again for children below 12, as you indicated that is yet to be approved, is yet to be authorized by the FDA. This is the emergency authorization again. So what is your recommendation for these children when they go to the school, other than personal hygiene, masking, and physical distancing, as you talked about?
There's really not much else we can do for these children at this point in time. Once the vaccines either get authorized or approved, I would obviously recommend that the children be vaccinated as quickly as possible.
Thank you. That ends my question. And thank you for the real succinct answers.
You're welcome.
Thank you, Amit. By the way, from the question you may have recognized, Amit has many years of experience with FDA. So thank you, Amit. Thank you, Dr. Chatterjee. We are going to go to next question. For next question, Robin Ghoshal. Robin, if you can unmute, Bhaskar, if you can unmute Robin Ghoshal, please.
Yes.
Robin, can you?
Yeah, I can listen to you. Yeah.
Do you want to turn your video or?
It's on. Is
Okay.
it?
Yeah, it's on. Robin is calling from Princeton. Go ahead, Robin.
Oh, hi. Dr. Chatterjee, it's excellent presentation. It is, you covered pretty much everything that we need to know. Still, I will be playing a little bit devil's advocate now. And I also listened to I participated in your advisory council meeting, and where you oppose the children, as Amit Mitra said, that you oppose that emergency use of authorization of the children because it has not been started. And now you think that it is okay to go with the children?
Yes. So I think it has to be put in context, your question. You are referring to the no vote that I made back on December 10th of 2020. So if you go back and actually look at, and I understand that you attended the meeting, then you know that at the time, the sponsor, Pfizer, presented data on 16 and 17-year-olds that were very small in numbers. They had data only on 150 children. And my no vote was really based on that number because I felt that it was insufficient data at the time to give authorization. The second thing was that at the time, the data we had, again, time is very important. So if you think back to December of 2020, most of the hospitalizations, deaths, the serious illness was occurring not in children, but in people who were over 65, who had major health conditions, etc. And so they were not considered to be at the same risk. The third thing is that we knew these clinical trials were ongoing. They had 2,000 children who were enrolled in the clinical trial. So what I wanted to see, which ultimately they did provide to the FDA, was the data on those additional 2,000 kids that were not available at that time. And so my no vote at the time was based on the paucity of data. Subsequently, we have seen that those studies have progressed. We've seen more data, and we have the efficacy and safety data in children over 12, and I'm satisfied with those.
And one more thing that
Robin, can you go with one question because we have a lot of people.
Sure.
And then we'll come back to you.
How we know? How we know who is the asymptomatic carrier, and how we can define asymptomatic carrier? Because I have seen people in the household of 10, one infected people comes in, nobody is affected, only one or two. How is it possible? Let's say in a room, how many cubic feet in 3,000 square foot house, 10-foot ceiling, how many volume of air that you have, the particle floating around? What is the viability of that to get infected? Otherwise, we have seen a lot of information about spreader or any mass media, mass kind of attendance. There will be effective. Somebody will get infected, and they will. What is the crux of that? Who will be the asymptomatic carrier? How we can recognize that?
It is impossible to do that because there are so many variables in there. It depends on the person's own health status. It depends how much of the virus they may have picked up from somebody else. You mentioning some of the factors, which is the size of the room, the number of air exchanges, etc. There are so many variables in there. It's absolutely impossible to tell. So this is why we say that from a public health standpoint, your safest bet is to get vaccinated. And even if you're vaccinated, given the increasing rates of infection that are occurring in the US, especially if you're in an area of substantial or high transmission, as the CDC calls it, it is wisest for you to be wearing a mask if you're indoors with other people where you don't know what their vaccination status is, or if you're in a large crowd.
Thank you, Dr. Chatterjee. Thank you, Robin. We're going to go, Bhaskar, to Deepak Chatterjee. I think he raised a hand.
Okay.
We see.
Thank you, Dr. Ghosh and Dr. Chatterjee. Thank you for answering. I think you have answered most of my question. One thing, in case we have the booster shot, does the brand of your vaccine that you have taken have to correlate with the brand of your booster?
Yes, it's a good question. And it's a question that actually we don't know the answer to, because as I said, we have to look at the vaccines as they were studied. Now, the vaccine sponsors, the manufacturers, will study their own vaccine. They're not going to boost with somebody else's vaccine. So I anticipate that if a booster is recommended, it will be with the same vaccine that you received before.
Thank you.
Thank you. Bhaskar, I have no way of knowing the order, but I'm in my list. I'm seeing Bishu Chatterjee. He had the first question. He's back. Can we turn him, and then we'll go to Deepali Banerjee and Monotosh Banerjee. I think that's the order. So Bishu, if you can hear, turn on. Something happened to Bishu. Go ahead. Bishu, you're unmute. Please unmute.
Is about immunocompromised people. What we have been hearing, from the media at least, is that the immunocompromised people, even if they have taken 2 shots, their immunity is still not there. And in fact, that people are saying that there is no immunity in spite of the fact that they have taken two shots.
No, that is not correct. That is not correct. And this is where misinformation gets transmitted. So there are very few actual studies that have looked at this population, but there is one that looked at kidney transplant patients, so solid organ transplant patients. And it is true that in that study, the vaccine efficacy was only 17% compared to the 90+% that you see with the normal population. So it is anticipated that in fact, the vaccine efficacy will be blunted. But in terms of data, we don't really have data from a lot of clinical studies and trials to share. And when people say, well, it doesn't work at all, that's actually not true. These are transplant patients who are on heavy immunosuppression. And even with that, they had some efficacy. And remember that we are measuring only antibodies. So we don't know if they have immune cells that work, et cetera. But it is just something to be aware of, that that is a vulnerable population, no doubt. And they need to actually use mitigation measures much more perhaps than the rest of the population does. So mask wearing, social distancing, the hygiene measures that we talked about earlier.
Is there any benefit from the booster shot for these people?
We don't know yet.
Okay.
That is being proposed, but we don't know yet.
Thank you, Bishu. We'll go to Dr. Deepali Banerjee. If Deepali or Bhaskar, if you can unmute and ask the question.
Thank you. Dr. Chatterjee, we really enjoyed your talk. Can you see me or hear me?
I can both see and hear you.
Okay, thank you. Okay, first of all, I just want to mention one thing. I think Deepakda already mentioned about if it has to be the same kind of vaccine for the booster. Now, I do have my cousins living in Toronto, and they had taken Pfizer first time, and then they had to wait a long, long time because there was a paucity of vaccines over there. But eventually, actually, I correct, they had Moderna first and got Pfizer the second time because they were told it's fine.
Mm-hmm. Yeah, and that does happen, as I'd mentioned during my prepared remarks, that has happened in some cases. Either the other vaccine is not available that you got before, or you moved to a different country or something like that. And the most important thing is to get that second dose if a second dose is needed. Ideally, you would get it with the same vaccine, but if you cannot, another vaccine which is very similar. So the two vaccines, as I showed you, are very similar. They use the same technology. The formulation of the vaccine is a little bit different. So the lipid nanoparticles, that fat bubble that they enclose the mRNA is different. But the mRNA is the same mRNA. It is not different. So it is the assumption that you will actually be able to boost. The reason we say don't do it is because we don't have data to support that. But if you have to, you don't have any choice, then it's better to get that second dose with another vaccine, which is similar, rather than not being vaccinated with the second dose at all.
Okay, thank you. My second question is a very, I believe this question is for everybody. We constantly get invitations for big Indian gatherings. And, there was a time when I started going, I went to two, after actually emailing to CDC, this was a wedding reception. This was before the Delta variant became so rampant, and they said, oh, you can go back to your pre-pandemic lifestyle. Okay, no mask, no social distancing. And then we were there for not too long. But then we came back and then boom, comes the big thing from WHO. If you can avoid large Indian gathering, that's what it was said. So I am trying to follow that. I feel very bad because I have very good friends who are having different gatherings there for a very valid reason, obviously. And so my question to you, if it's indoor, outdoor, is there a difference? And number two, what should we do?
Yeah, so there's definitely a difference. And we know this, and the CDC has actually been very clear about this for a long time now, to say that outdoor activities are much less risky than indoor activities when it comes to spreading this virus. So if you're outdoors, even if there is a larger group, that's probably safer than if you're indoors. Even so, I mean, I live in Chicagoland, and we had, Lollapalooza, which is a huge event that took place in Grand Park, hundreds of thousands of people there, shouting and screaming, et cetera, which of course we know spreads the virus, right? So even if you're outdoors and you're standing next to someone who is infected and they're yelling and screaming, you're likely to pick up that virus. So I think you have to use common sense when you are making these decisions. I would also say that when there are events, like you say, weddings or other types of social events that people are having in their homes and things, I think people need to be very understanding and tolerant. When someone says, I would love to come, but I don't feel safe, I think it's fine, and people should take care of their own safety. It doesn't mean that we become hermits, but it does matter, and we have to be thoughtful when we are deciding whether to accept an invitation or not. I will tell you, we had two couples who came over for dinner at my house last night, okay? And we're all vaccinated. We know that the risk of transmission within this small group is pretty small. They felt comfortable coming. We felt comfortable having them over at our place. I can tell you that I see a couple of our friends from Omaha have joined, Moa and Shanit Ray. They would come to our home in Omaha. We would have 50, 60, 100 people in the house. I would not have those kinds of events in my house now because of this risk of transmission. So I think you have to know where you are, who you are with, what are the circumstances. And I think people need to not feel guilty that you're not attending something because you're trying to keep yourself and your family and your friends safe.
But if I want to go indoor, do I have to wear a mask all the time? The problem is when you're eating.
Yes, so that question has been asked and it actually has been answered. This is a change. The CDC did initially say that you did not need to wear masks if you were fully vaccinated. If you're with other fully vaccinated people, they have changed that because of actually an outbreak that occurred in Massachusetts. There was an event that was held with 400 some people. It was a 4th of July event, and 300 some people got infected. They were not all very sick. There were a few people who got hospitalized, but nobody died. But they became aware through that even in a highly vaccinated population, the virus can spread. And so that's why the recommendation is, particularly with the current rates of spread that are occurring, that even fully vaccinated people wear masks indoors if they are in larger groups or they're with people whose vaccination status they don't know.
But what happens when you're eating?
Yes, so that is a very difficult situation. And that's why I said that smaller groups are preferred where you can actually socially distance while you're eating. It becomes very difficult when you've got large numbers of people to be able to do that. So Again, use good judgment when you are deciding whether to go to these events or not. And it's perfectly fine to ask, who else have you invited? How many other people are going to be there? And make your decision based on that information.
Thank you, Dr. Chatterjee. Appreciate it.
Thank you, Deepali. Thank you, Dr. Chatterjee. We're going to go to Dr. Manotosh Banerjee, waiting long time. Manotosh, go ahead, please.
Yeah, Dr. Chatterjee, first is that we are so fortunate that we have people at a very high level attending here who are part of FDA and have got a authoritative information. And your slides are absolutely very right on, not too deep, but still adequate for understanding. My comment is related to the fact of what Deepali said, the wearing of mask. One clarification is never mentioned. Mask is not intended to prevent virus from entering through those cloths, because masks in medical setting are usually for protecting others. Surgeons have worn masks for 100 years. They have been not wearing to protect themselves majority of time, unless the particle was so large, or a respiratory bacteria like TB, for which you may have an N95 mask. But cloth mask does not protect you even from TB nearby. So it cannot protect a virus from virus. So mask is more for protecting others from the droplet from your mouth, and which can be trapped by a cloth mouth effectively, but not for protecting oneself. So mask, again, is never used for protecting oneself. Now you can put 6 layers of mask.
I agree with you on that. I don't think that is actually correct.
No, but I'm saying that if you are nearby and somebody's droplet comes in there, it will be protecting you.
No, because I will tell you something very simple that people don't realize. I'm an infectious disease specialist, right? I've been practicing for more than 20 years. I see patients with measles, with TB, with whooping cough, okay? When we enter those rooms, TB is a special situation. As you mentioned, you need a special kind of mask, the N95 mask for that. It's actually a respirator because TB spreads in a different way. But for these respiratory viruses, if you enter a patient's room who has flu, we would wear PPE, including a mask. That's not to protect the patient from our secretions. It's to protect us from becoming infected with whatever the agent is in that room. So actually masks do provide protection to the wearer.
But I meant only cloth mask. I was talking about cloth mask, not a full PPE. I'm not talking about a mask, shield, etc. Yeah.
So it's a good point you make, the difference between the different types of masks, because it is true that cloth masks are not nearly as effective as other masks are. We don't have time to go into the relative efficacy of different types of masks, but I can assure you that any kind of nose and mouth covering will provide some level of protection, and people should be using them. They are not as effective.
No, but mostly for the person wearing or protecting others. That's my point.
Both ways. Both ways.
Which one is more in your assessment? Protecting others or myself?
As I said, there isn't enough time to go into the details of the mask, because those studies have actually been done in terms of the particle size. There are multiple variables. It's not just cloth or
I agree.
Yeah. So you need to keep in mind, something is better than nothing. The surgical masks are better than the cloth masks, and the N95s and N95s are better than the surgical masks. That much I can tell you.
Other thing is that those people who are fully vaccinated against alpha variant have a gap that you showed in your slides, and they were not head-to-head study. And these efficacy numbers are for serious disease only and death. So 95% efficacy is for serious disease.
No. 95% efficacy is for symptomatic disease. It's not.
That's right. Okay, but the people can have asymptomatic disease.
Sure. They can be asymptomatic carriers. That has been known from the clinical trials. That has been known for a long time. I agree with that.
I just wanted to go on.
where
Yeah, no, I
they,
got your, no, I got your.
where they actually asked this question. I got asked this question way back
Sure,
in
sure.
January
Go ahead.
as to why the studies were not done to prevent asymptomatic carriage. And that is a complicated vaccinology question. These types of vaccines do not prevent nasal carriage. And it's very difficult to actually make vaccines. We do have vaccines that prevent that. The measles vaccine, the chickenpox vaccine, they do that. But those are live virus vaccines. Those are weakened live virus vaccines. We don't have any such vaccines against COVID.
Yes. And if I am already Delta variant infected, I may not know that because I have been vaccinated by Alpha variant and my symptoms may be less. So again, my concern is to protect others. And that's why I insist on wearing mask.
Good for you. So actually you were not vaccinated with the vaccine that contained the Alpha variant. What you were vaccinated with was before the Alpha variant.
That's
before
right.
any of these variants became.
Yeah.
That was the original version of the virus that came out of Wuhan, China.
Yes.
We're
Thank you very much.
Thank you, Monotos.
Thank you.
There are a number of people waiting. We'll go to Dr. Shanti Banerjee. Shanti Babu, if you can hear, unmute, or you already unmuted. Go ahead.
Yeah. Well, excellent presentation. Thank you. I have a question which is different. We get a lot of side effects. You said your daughter got it. And my question is that side effect generated from impure mRNA? Or how is this mRNA generated?
The mRNA doesn't generate anything. The side effects that you're talking about are generated because of the immune response. So that is why you see the difference between the first dose and the second dose, right? Because you're giving the same vaccine, but you're seeing the reactions happening after the second dose because your body has already developed some immunity to it. It's now seeing a target and it's reacting. And so you're having the symptoms like fever, headache, muscle aches, etc. That is because your immune system is recognizing that vaccine as something foreign. And it's actually, the vaccine is working. That's why you're having those reactions.
It's not due to impurity.
It has nothing to do with the actual
messenger RNA.
No, no.
And the another question is when does this mutation occur? During the passage through the body?
No, so it's a very important question you ask, and that is that these mutations actually are occurring as we speak. There are thousands of mutations that are occurring because this infection is so widespread across the world. These mutations are occurring continuously, and some of them become more established, like you heard, the alpha, beta, gamma, delta variant, etc. That's because they have a survival advantage over those other mutations, which are not able to survive necessarily as well. So the mutations actually are occurring continuously. And when you talk about mutations within a single person's body, that actually has been demonstrated. In an individual who was immunosuppressed and carried the virus for several months, they were actually able to get a series of samples from that patient and find the mutations as they occurred. So they can occur, but in most people who don't carry the virus for that long, the mutation may occur and then it gets passed on to somebody else. It's not continuing within their body. Did I answer your question correctly, Dr. Banerjee?
Yeah, but isn't the virus, ineffective or dead outside the body? It becomes active inside the body.
It's not dead. The virus simply gets transmitted and changes the host. So it goes from you to another person, from another person to you. It is not dead anywhere along the way.
But as long as it is in the air, my understanding is the virus is not active.
It can survive for a very short time in the air. And yeah, it's not active because it has to have a cell in which it's multiplying in that, and that's when the mutations occurs, when the virus multiplies.
Thank you, Shanti Babu.
Okay, thank you.
We have a number of people.
Excellent presentation.
Thank you. We are going to go to Shravani Chatterjee. Shravani, if you can unmute.
Yeah, hi, Dr. Chatterjee. I had question because I have a daughter who is below 12 and who is not vaccinated, right? And the school is saying that from fall there is no remote option, it's all in person. And then this whole Delta variant has come into our life and it's becoming worrisome for the kids who are not vaccinated. So is there a plan for kids below 12 is a vaccine? Because I saw in some media house that's first quarter of 2022. That's kind of late for these kids. I don't know. So if you could explain to us why this is getting delayed, or is there any plan for them?
Yeah, so it's not actually being delayed. The strategy that the vaccine manufacturers have used is to go in reverse order for age. So they started with the oldest kids first, the 16 and 17-year-olds, because they're similar to adults in terms of their immune systems. And so they were expected to have very similar responses. So they started there. Then they went to the next age group, which was 12 to 15-year-olds. The next group is the 6 to 11-year-olds, the school-age children that you're talking about. After that are the 2 to 5-year-olds, and then the 6 months to 2-year-olds. So there is this reverse order in which they are doing the clinical trials. I am very wary when people say, well, we'll have a vaccine by December, we'll have a vaccine by first quarter of 2022. I don't know on what basis they're doing that. I think a lot of it is guesswork, and I don't engage in guesswork. I only wait to see once the data actually become available through the FDA, then we can say, yes, we are likely or unlikely to have that. But until we see that, there are many, many problems that occur in clinical trials. As Dr. Ghosh told you, I've conducted 120 clinical trials myself. No one can give you guarantees of when clinical trials will be completed. Having said that, I recognize your concern for your child, and a lot of people are worried about their children. As I mentioned, there's some simple things you can do. Children are very smart. 2-year-olds can actually wear masks very, very effectively. You know, people said, oh, they're going to pull them off, you know, they're going to exchange masks and do all this stuff. You know what? They're not doing that. Those studies have been done where they've looked to see, can children wear masks all day in school and actually learn? And they can, not just in the US, across the world, they're able to do that. So I think if you teach your children the importance of wearing masks, and importance of hand hygiene, et cetera, that they are actually going to be able to keep themselves safe as much as possible. I think the learning is very, very important. Dr. Ghosh mentioned, I'm a member of the Pediatric Infectious Disease Society Board of Directors. We actually supported the statement that came out from the American Academy of Pediatrics. It's supported by a lot of infectious disease societies, the Society for Healthcare Epidemiology of America, to say children need to be back in schools. They need that learning environment. A remote environment is not adequate, particularly for young children. We know that they have developed educational gaps. They've developed gaps in their social and psychosocial development. It is very important for children to go back to school. Now, a couple of other things I will say, that the schools should be requiring those who can be vaccinated to be vaccinated. So faculty and staff should be vaccinated. They should be able to socially distance where that is feasible to do. And then the last thing that I will say, and again, it is just to reassure people, if you're hearing these media reports that there are increasing cases among children, well, that is because we have stopped the transmission in older people. So the proportion of cases in children is more. But most children who become infected actually do not become very ill with this. Most are asymptomatic or mildly symptomatic, and they will recover from this infection, even if they have it without too many long-term side effects. Does that answer your questions?
Yes, yes, yes. Thank you. It assures, because we are reading so many things, it assures from a person who is in this actually, who's doing all the studies and everything. Thank you very much, Dr. Chatterjee.
You're welcome.
Thank you, Shravani. That was excellent question. Dr. Chatterjee, I have received before your presentation, number of questions related to almost the identical question that Dr. Mitra in the beginning and now Shravani is asking, because that's a very worrisome question. In my family, I have the same situation. My daughter has two 7 and 4 years, and another month or less than a month, both of them will go back to school. it's almost mandatory. So we are worried. So thank you. I think that's a good assurance. We're going to go to Jayanto Mukherjee. He's patiently waiting. Jayanto, if you can hear, go ahead, please.
So thank you very much, Archana Di and Shamodda, so for joining this meeting. And also, I just want to tell you that they were member of BAGC for a few years. They moved here and they were member. And very recently, as a honorable guest advisor, they have joined our executive committee of 2021. So we are glad to have them in our committee. So this is a really excellent session. Many knowledge we got, and I think our community needs all these questions and some answers like this, which clarified a lot of doubts we are having. And from the executive committee also, we are having also some situation which we need to mitigate. And I hope Archana Di will help us on these things. So we are going to have some in-person event coming soon, like August 28th, and then Durga Puja, Kali Puja. So how do we mitigate those things? So the thing is that we can always make the mask mandatory, that we are going to do it. And we can actually give surgical mask, double mask to everyone. Now the situation is with the kids below 12 years old. How do we handle them? Because they can, there can be some people with vaccination may carry the virus. So there is a suggestion made by a few other companies outside that do a COVID test on site. And there is a COVID test available for like a 15 second, 20 second. They give you an instant COVID test. And even for the 2-year-old baby, they can do a saliva COVID test. So that has been recommended to us that even for vaccinated, non-vaccinated, you do a rapid test before you let them enter the indoor space area. As long as you are in the outdoor space area, you are okay. The moment you enter the indoor area, we do that kind of mitigation situation that we do an instant COVID test. So it will be for the vaccinated people also. That will make sure that even a vaccinated people coming in without any infection. So we are thinking about that. Don't know actually exactly how to implement that, because testing all those people in a short period of time in an event will be a very, very difficult process. And definitely we are going to force everyone to put double or single surgical mask. That's what we can. N95 we cannot probably afford, but anybody wants to do N95, we will do it. So these are the things we are thinking, and Dr. Chatterjee will definitely help us. And also thanks to Shamodda, he has been helping us already in our Bongo Bhavan situation. He is a very senior structural engineer. So we welcome to them in BAGC. It's very nice to have them with us. So thank you very much for everything.
You're welcome.
Thank you, Jayanto. We have a question from our technical team, Bhaskar. Bhaskar, go ahead, unmute yourself. Bhaskar,
Dipankar Da has a question. Shall we go there first?
Dipankar Da, go ahead then. Then we'll go to Bhaskar. Dipankar Da, if you unmute.
I thought I saw him and then he disappeared.
Yeah, I saw the hands. I thought.
I don't see him anymore.
Okay, why don't you?
He's here.
Can you switch on his video then? If you're okay. Okay.
Yeah.
Shankarda has also raised his hand.
I can try.
Dipankar was there with the hands. I saw.
Yeah, he's there.
I see Bivasda here. Bivasda, you're welcome to come and ask question. So
I unmuted him.
Okay. So Dipankar Da, can you hear us?
Yeah, I can hear you.
Somehow, can you switch on your, if you want, you can switch on your video. Up to you.
It says, okay, you cannot start your video because the host has disabled it.
Can you try once more, please?
There you go, Dipankar Da. Go ahead, Dipankar Da. Dipankar Mukhopadhyay.
Hello, Archona, Dr. Chatterjee, from a fellow pediatrician, much older. I had a couple of questions. One was, in the Pfizer statistics on mostly rheumatoid arthritis patients on steroid or other biologicals, they had originally a series of 130 patients. Later on, they had about 330. And there, what I remember reading, that if you had one dose of Pfizer, you were 48% made antibodies. And if you had two doses, you made 64% of the time antibodies. And as I understood also, nobody still knows which antibody is protective and which is not.
That is correct. It's not that we don't know which antibody is protective. These are neutralizing antibodies, so they are protective. What we don't know is what the serologic correlate of protection is. And what that means for the general audience is what level of antibody you need to protect you. You know, is it 100, 500, 1000? We don't know that. And we actually don't know that for many other diseases. So for example, for whooping cough, we don't know what the serologic correlate of protection.
Yeah, I understand that.
So
Nature had an article.
But we do.
Nature had an article that in bone marrow they found the plasmacytes and so on, and keeps the virus longer, and they can actually start working on it with reinfection.
So the problem with people who are immunocompromised, immunosuppressed, as I mentioned, whether they are receiving biologic agents for rheumatoid arthritis or the autoimmune disorders, whether they're getting chemotherapy, whether they have a transplant, the level of response to the vaccines varies depending on the level of immunosuppression. So I had mentioned the kidney transplant patients because they are the most immunosuppressed and they have the lowest level of protection. It varies depending on what type of immunosuppression is being used. There are lots of variables here in terms of the patient's status, how long is it since they got the immunosuppressant, etc. So it's very difficult to tell exactly in an individual patient whether they will respond or not respond to the vaccines. At the moment, the recommendation is to vaccinate them all, because if there is any hope that they are going to have some response, they should be vaccinated.
Right. My other question was for adolescents who had reaction to the second dose. If they want to go to contact sports, should they have a cardiac evaluation?
There is no such recommendation because, as I had mentioned before, the risk of myocarditis or pericarditis is very small. And these are not sudden cardiac events. These are actually symptomatic patients who will have symptoms. So of course, if they have symptoms, they need to be seen by a physician, and they shouldn't be going to any events if they are symptomatic.
Right. So basically, these are not silent lesions.
No.
Thank you very much.
Thank you.
Thank you very much.
We'll go to Shankar Sarkar. Shankar, please unmute.
Can you hear me?
Yes.
Yes. Thank you, Dr. Chatterjee. Before I ask my question, I just would start off with just a comment saying that currently we have the alpha, beta, gamma, delta, eta, epsilon, iota, kappa, and zeta variants.
There's also a lambda.
There's a lambda that also surfaced very recently. So fairly soon, we'll be running out of Greek alphabets for nomenclature. that was my first comment. Now, let me also say this. In the context of the natural evolution of the virus, what trajectory are we to expect? Now, let me say this. The Spanish flu of 1918, it waxed and waned over some years before it died out as people developed herd immunity. I mean, there were no vaccines, there was no tracing, very, very little understanding, obviously not sufficient vaccines, etc. So, and the human—
there were no vaccines. The only thing they had were mitigation measures, which they used. So lockdowns, social distancing, masks were used.
Yes, quarantine, etc. So the human toll, eventually it was staggering. I mean, I think currently the worldwide toll stands at something like, it's just a shade below 5 million.
That is correct.
but in the larger global context, in the Spanish flu, the statistic says that anywhere from 50, almost 50 million people died. I mean,
that's correct. That is the estimate.
Yes, that was the estimate. So it was staggering. So the literature actually says that even today, that there are some residual strains of the Spanish flu which
that is
are
also
rather
correct.
inconsequential.
Yes.
Yeah. So do you think, I mean, again, this is conjecture, do you think COVID may follow a similar path? I mean, we are talking about, PPE. So even as we play catch up, we are talking about booster shots. We are talking about the Delta variant. Now you have the Lambda. So there is no telling. I mean, there may be another particular variant that may be much more virulent and much more damaging.
That's entirely possible. That's entirely possible. It's very difficult to say whether that will or won't happen. What I am sanguine about is that this virus has now been around for a year and a half, and we haven't seen that happen. So what we know about viruses is that if they were going to develop that kind of a variant that would become, what we call escape mutants, you got a mutation which would escape everything we have against it. That's possible. That's not likely, in my opinion. The doomsday people like to talk about that, but I'm glad that you put the COVID pandemic in context with the 18-19 pandemic, because I think that's a scale difference that people don't understand, and it's a very important difference to understand. This virus is dangerous, no question, but it's nothing compared to what that influenza virus was.
Yes,
and there have actually been other pandemics that have occurred in the past where, for example, with the Black Death in the Middle Ages, a third of the population of the world was wiped out, which people have not realized. So we are nowhere near that. And I think what we need to be guided by is the modern science, information, data that we have that is accurate, and not to sort of go down these rabbit holes of conjecture that people make.
Yes.
Thank you, Shankar.
Thank you. Again, the point I was trying to make is that there is a certain randomness, and we are always playing catch up because we are reacting to certain sets, occurrences across the world. So, I was thinking that maybe, it would have to come from herd immunity eventually.
Yes, eventually that's what happens. A sufficient proportion of the population becomes immune either through vaccination or through natural disease, where the disease transmission can no longer occur and the virus dies out.
Okay, thank you. Thank you, Shankar. We're going to go for one last question from Bhaskar. Bhaskar, please go ahead and ask your question. We are just about the point of 1 hour, 30 minutes.
I think Banidhi also wanted to ask question. Banidhi, do you want to?
As an engineer and as a man of science.
So let me, since I have the mic, and in the interest of time, I'll just go ahead. Thank you, Archana again. She's been very kind enough to spend the time, and I will want to remind, we had a podcast recorded for an hour where she takes us through the evolution of the entire vaccine. So one quick point, if you, and we can take it offline as well. I have been seeing, recently articles on monoclonal antibodies and its authorization. So however you want to explain that for us would be really beneficial.
I'm going to do my best. I'm not the expert in monoclonal antibodies, but these are what we call render passive immunity. So when you get a vaccine, your body makes an immune response that's active immunity. When you get a monoclonal antibody, that's a pre-made antibody that is given to you. And monoclonal simply means that it is made in the lab. So it's a very targeted, it attacks only one specific epitope, one specific antigen on the virus. That is what monoclonal means. Most of us heard a lot about this, especially when President Trump came down with COVID and he was treated with different cocktails and things like that. People heard a lot about it. The thing with these monoclonal antibodies is that they are very expensive to make. They are effective only against that epitope. So if the virus changes, the monoclonal antibody is no good. So you've got to make a new one now. So that's why they're difficult to use, and they are reserved for those reasons to be used only in people who are fairly sick, and they also work only in the early stages of the infection. So once that immune response has started within the body, and the person is already having a lot of symptoms and in the hospital or in the ICU, they're actually ineffective. So if you look at the studies, some of those don't look very good because they gave the monoclonal antibody too late. You have to give it early on in the infection so you can stop the virus from spreading within the body and causing the havoc that it does once it takes hold.
Thank you, Bhaskar. Banidhi, do you have a question? If you do, you'll be the last one. Go ahead, Banidhi. Dr. Bani Bhattacharya.
I do not. I'm an anesthesiologist for long time, but I really liked your lecture. It's very informative. I don't have question because I think whatever you said, I knew most of it, and I do not have any question so far. I can remember. But it was excellent presentation. And
thank you, Banidhi.
Thank you, Banidhi, for your comment. I think we're going to wrap. I think we try to wrap within an hour and 30 minutes. We are just at that mark. Like to thank Dr. Archona Chatterjee once again for your valuable time for last hour and a half. And I'm sure many hours you spent before preparing for these slides. So thank you. And also we are very proud on behalf of Bengali Association, some of the services you and your husband joined to mention you are providing. So we are grateful, we are thankful. We are really will look forward to work with you.
Thank you very much, Dr. Ghosh. It was my pleasure to speak with this audience tonight, and we are delighted to be part of the BAGC community.
Great, thank you. I'd like to take this opportunity to sincerely thank our technical team, Bhaskar in particular, and Jayanto. You don't know how many hours they spend behind the scene and during the seminar. So thank you, technical teams. There are others. Sravani is part of it. Jayanto is part of it. So thank you all for helping in this seminar, monthly seminar. It's once a month. This is our third year we are doing. Unfortunately, COVID made it difficult. So now we are doing virtually. At this point, Jayanto, do you have a word or two for concluding remarks before I close?
Just one thing. So we actually made this program live in our BAGC Facebook and also our BAGC live YouTube channel. The whole presentation is going to be staying there. And if you want to share that link to any of your friends, you are welcome to. I see that Vivasda has asked a question. So if he has any further question, he can ask still now. Another thing is that we also recorded this in the background. And one more thing, Archana, you mentioned that whatever slides you presented, if anyone wants the copy of it, we can share to them. I don't want to share to the whole BAGC Google group members. So in case you need it, please request to us and we'll share that to you.
Thank you, Jayanto.
And thank you very much, Deepak, for organizing these events for us. And this was a really awesome event and very informative and timely on the current situation. And this has helped us clarifying lots of our doubts. And thank you, Archana. Thank you.
Okay. Well, that concludes our presentation today. Once again, thank you, the BAGC members, for your active participation. Without you, without your encouragement, there won't be any monthly seminar, particularly very informative ones like this one. So thank you all. Good night. Stay safe. As Dr. Chatterjee said, take precautions. There are a lot more unknown that we know at this time. So why not take the precaution that we can control? With that, thank you. Good night. Stay safe.